but there are detailed medical records on how he perceived the speech of others. In Henry G's own words, "I cannot get it. It does not finish. It's one on top of the other. The words split themselves up. The noises are not quite natural. I hear your voice, but not the words. I can hear, but not understand." Henry G had a rare condition called auditory verbal agnosia, or pure word deafness. In this lecture, I'll talk about how disorders like this one shed light on the neural mechanisms for language comprehension. The story I'll tell highlights all three of our neural principles: specialization of function, the network property of the brain, and the power of plasticity. To better understand the journey that speech takes to create linguistic meaning in our heads, I want to start with a different modality: vision. Vision is one of the most studied processes of the brain, and neuroscientists believe that some fundamental aspects of vision are shared by many other systems, including language. When a photon reaches the eye, it gets chemically transduced into electrical signals that are sent through subcortical relay stations to the back of the brain in the occipital lobe. By now, you know that the primary visual cortex, or V1, is located in the occipital lobe, and that's the start of the two journeys I want to describe. From V1, visual information gets split into two parallel streams, one going downward and one going upward. The one going down is called the ventral stream, and its function is to process the visual details of whatever thing a person is viewing. So if the object before you is a snake in the grass, the neurons along the ventral stream increasingly analyze the details of that snake. The farther down the stream, the more specialized and accurate the processing. This detailed processing would allow you to determine what type of snake it is. Is it harmless or is it deadly? The neuroscientists David Milner and Melvin Goodale named this pathway the what system, as in, what am I looking at? At the same time that this is happening, the dorsal stream is processing a complementary piece of visual information: the location of the object. This pathway runs upwards from V1 through brain areas specialized for processing motion and finishes up in the parietal lobe, where neurons analyze the relative location of the object to one's own body. This pathway is often referred to as the how system, as in, how am I supposed to interact with this thing? In the case of a deadly snake in the grass, the answer is probably get back. These systems work in parallel to process visual information, and given what you've learned so far, you might not be surprised that damage to one part of the system impairs one aspect of vision, but not the other. So damage to the front end of the ventral stream disrupts the ability to correctly identify what an object is, but spares the ability for the dorsal system to determine where it is. The opposite is also true. Damage to parts of the dorsal system make it impossible to detect motion or location, but spares the ability to identify objects. Interestingly, there's even an unusual condition called blind sight, where all of V1 is damaged, but because there are ancient pathways that bypass V1 and enter the ventral and dorsal streams subcortically, these patients can't consciously see objects, but they can successfully guess their identity and location. Remarkably, when asked how they were able to get it right, these patients report that it was just a hunch. Neuroscientists believe that the primary function of having these two parallel visual processing streams is that vision can specialize its processing while also ensuring that it's very fast. Evolutionarily, inefficiencies in the visual system would have been strongly selected against. A visual system that first had to identify an object before processing its location, or the other way around, would have been much less adaptive than a system that does both in parallel. Perhaps the strongest evidence of this adaptiveness of this particular dual stream system is that it is evolutionarily conserved in many other species. We're not the only ones on the planet to have evolved this nifty strategy. This neural strategy of dividing and conquering is so effective that we now think it is also used for language processing. Although originally hypothesized by Carl Wernicke himself in the late 1800s, this dual stream hypothesis for language has gained new life a century later with the advent of functional neuroimaging methods. Most notably, a version of this model was reinvigorated in 2007 by the cognitive neuroscientists Gregory Hickok and David Popple. For the remainder of this lecture, I will sketch out the two main parts of their dual stream model for language comprehension. But first, a caveat. We are just beginning to piece together this incredibly intricate network. Unlike researchers on vision who draw extensively on non-human models, neuroscientists studying language are stuck with humans because we're the only ones who have language. And now, in the golden age of neuroimaging, we are finally able to tell more complete stories and more complex stories about language than ever before. There are still many debates over the details, but I'll present the basic framework of what we know. Have you ever wondered why your ears are folded and grooved like they are? It's not for looks. The shape serves a very specific auditory function. These folds and grooves are perfectly designed for capturing sound waves in the frequency range of 500 to 4,000 hertz. Hertz are just sound wave cycles per second. Do you know what type of sounds in our environment mostly happen within that wavelength? You guessed it, human speech. When sound waves travel towards us, they get funneled from the external ear, called the pinna, inward to the eardrum. Once the pinna funnels and amplifies the speech sound waves, they hit the eardrum. The vibrations there are mechanically transduced into action potentials in the cochlea, which are then sent via subcortical relay stations to A1 in the temporal lobe. As I mentioned in an earlier lecture, A1 is topographically organized for sound input, just like V1 is for visual input. That means that there are cells in A1 specialized for different sound frequencies. As with V1's retinotopic organization, the primary function of this tonotopic organization is to allow efficient and sharp contrasts among the sounds. This ensures that the sound gets accurately sent to the next stage of processing. From A1, all processes move to the surrounding superior temporal cortex, which has been called traditionally Wernicke's area. At one time, researchers thought that this large area's main function was to attach meaning to words, but we now know that it has multiple functions. One of the earliest stages of processing after A1 is to recognize acoustic features of words. It was once assumed that this processing occurred only in the left hemisphere, but we now know that it is bilateral. For example, neuroimaging scans show that patients with lesions to the left superior temporal lobe, with the right homologue intact, can still recognize words as words. However, bilateral damage to this part of the brain is devastating for word recognition. In fact, this is most likely what caused Henry G's pure word deafness. Recall that he could not hear words as words. For him, it was all gobbledygook. And modern-day neuroimaging scans show that the culprit is likely to be bilateral damage to this brain region. And consistent with the fact that Henry G could process other sounds just fine, researchers have used fMRI on non-brain damaged participants to show that this region may be specialized for the sounds of language. Many studies have now found that the bilateral superior temporal lobe is more active for speech stimuli than complex non-speech sounds, suggesting that it is indeed one of the first processing points that treats speech as a special type of acoustic signal. Not only that, but this is also the first stage of processing that distinguishes phonological features within spoken words themselves. So this is where the brain distinguishes between minimal phonetic pairs, like rake and lake, or shujin and shujin. In keeping with the theme of dividing and conquering, this processing is bilateral. Hickok and Popple argue that the left hemisphere analyzes the fine-grained details of these phonological forms, whereas the right hemisphere does a more holistic analysis. This research has been further supported by Meg's studies showing that the timing of these phonological distinctions occurs roughly around 100 milliseconds after sounds hit the eardrum. This means that within 1/10 of a second, this bilateral superior temporal system can distinguish among the phonological forms of every word within one's language. Processes that occur this quickly in the brain are usually highly specialized and crucial for survival. Think back to a time when you struggled to hear the phonemes in a foreign language. If it was always that hard to process your native language, you'd be in a lot of trouble. From the superior temporal lobe, the two streams split and one heads ventrally and the other heads dorsally. These two pathways ride along two well-established white matter tracks. Recall that white matter pathways are like the information superhighways of the brain, and this infrastructure is what allows the brain to function as a network of specialized mechanisms. And here's an interesting fact, courtesy of the psycholinguist Julie Sedovy. It's estimated that the average 20-year-old has approximately 100,000 miles of these white matter tracks. That's a lot of road in our heads. Let's start with the ventral speech stream. The transitional area connecting the posterior superior temporal lobe and the middle temporal gyrus is where words are recognized as having meaning. Damage to this area produces receptive aphasia, which was traditionally called Wernicke's aphasia. Unlike patients with pure word deafness, these patients can produce and hear speech as speech, but the speech lacks appropriate meaning. So these patients may hear the word key, but they may think it means something totally different than you or I think it means. Interestingly, these patients will still be able to interact with keys in the appropriate way, so it's not as if they have no concept of a key. It's just that the link between the word and concept is broken. Long before neuroimaging methods corroborated claims that Wernicke's area was a mechanism for attaching meaning to words, Wilder Penfield observed this function directly in non-aphasic brains. Remember Penfield? He was the American-Canadian neurosurgeon who directly stimulated the brain with electrodes before he did surgery for severe epilepsy patients. He was the first to directly show that electrically disrupting part of Wernicke's area caused severe language comprehension deficits in patients on the operating table. With the advent of spatial neuroimaging methods, we now have literally hundreds of studies